Okay, so I think we'll get started. Welcome everyone to today's ImmuZoom seminar. So it's really my pleasure to welcome today's speaker, Dr. Wei Hu from Yale. I want to have a special thanks to Wei. She really saved the day, and our speaker has some health issues, and then she really jumped out and filled the spot. I really appreciate Wei's delivered today's lecture. So Wei currently is a fresh new assistant professor in Yale. His lab is part of the Department of Immunobiology and the Institute of Biomolecular Design and Discovery at Yale University. So Wei completed his undergraduate study in Peking University in biological sciences, and then continued her PhD training in Southwestern Medical Center in University of Texas. And then afterwards, she moved to Memorial Sloan Kettering in New York as a postdoc training with Sasha Rudzansky. Wei's lab currently focuses on developing and utilizing novel genetic, biochemical, and then chemical biological tools in combination with high throughput sequencing and then bioinformatics to study the differentiation and function of T lymphocytes. Her lab actually particularly study how immune responses are regulated by regulatory T cells, which is a dominant cell population suppressing a wide range of immune effectors. I think a lot of the audience probably already know. And then Treg cells has been also implicated in tissue repair and then regeneration through direct interactions with non-immune cells. And then which currently Wei's lab is focusing on understanding universal and tissue-specific machinery which regulate Treg biology, and then which could help in conceiving better strategy for targeting Treg cells for therapeutic purpose. And some of the ongoing project in her lab focus on elucidating the biochemical, molecular, and the cellular basis of immune regulation by studying common and context-dependent regulations of Treg gene expression and function. I think, as I mentioned, Wei's being trained with Sasha Rudzansky and then a very productive postdoc training, which actually result a series of high-profile publications, and obviously, she being received numerous awards, which including Elliott Goldening Awards, Immunogen Postdoc Training Fellowship from Cancer Research Institute, and then Ray Owen Young Investigator Awards from Midwinter Conference of Immunologists. So I think without further ado, yeah, we are very much looking forward to your talk, Wei. Thank you so much again for delivering the seminar today. So please go ahead.
Okay. Thank you, Tran, for that very, very kind introduction, and for giving me this opportunity to share some of our published and ongoing work and some of our thoughts on how Treg cells function in vivo, and how we're attempting to use new tools to get to the bottom of how they function at the cellular and molecular level. So probably no need of much of an introduction to this particular audience. We're all familiar that the vertebrate immune system can be partitioned into the innate and adaptive compartment, and there's a constant bidirectional flow of information between the two. For example, cells of the innate immune compartment, for example, dendritic cells, upon sensing microbial insults through pattern recognition receptors, such as toll-like receptors, can deliver activation and differentiation signals to effector T cells. And depending on the nature of these activation signals, the activated T cells can differentiate into different flavors of T effector cells. And these are known as the type 1, type 2, and type 17 effectors. And each one of them has a distinct cytokine profile that enables them to control and protect different classes of pathogens. On the flip side, uncontrolled activation of all three of these effector lineages can also lead to different types of immunopathologies that can end up being detrimental to the host. So some mechanism has to exist to make sure this whole process is kept under control, where the host can benefit from the protective effects of these cells without suffering from the immunopathologies resulting from the overactivated immune effectors. And one of the major mechanisms of immune suppression is carried out by a specialized T cell subset known as regulatory T cells, or Tregs for short, which my lab studies. So Treg cells are able to talk to and interact with a wide array of immune effectors and dampen their activity. And this includes direct suppression of effector T cells, as well as indirect suppression of T cell activation through the antigen presentation cells. Treg cells can also suppress antibody production by talking to the B cells directly. And last but not least, Treg cells can also suppress the activation of innate immune effectors, including NK cells, macrophages, and granulocytes. So Treg cells are characterized by the expression of this lineage-defining transcription factor called Foxp3. Foxp3 is an X-linked gene, which is essential for both the development and differentiation and functionality of regulatory T cells. And this is nicely backed up by genetic evidence in both mice and humans. For example, mice that are deficient in Foxp3 develop this early onset fatal systemic autoimmune disease characterized by lymphadenopathy and splenomegaly, autoantibody production, again, highlighting overactivation of the B cell compartment, immune infiltration and tissue destruction in various non-lymphoid organs, including the lung, the liver, and the skin, hyper IgE syndrome, as well as massive cytokine storm that spans type 1, type 2, and the type 17 flavors that we just talked about. Similar to the mice, there have been human patients that lack this critical gene or express loss of function mutations of this important gene. And these patients are collectively known as immune dysregulation polyendocrinopathy enteropathy X-linked, or IPEX for short. And these patients have clinical manifestations that mirror what is seen in the Foxp3 deficient mice. So all of these evidence show that Foxp3 expressing Treg cells are absolutely essential for preventing the onset of autoimmune inflammation resulting from overactivation of autoreactive T and B cells. So not only are Tregs required to prevent the onset of early of autoimmune activation at an early age, they're actually required throughout life to keep these autoreactive cells in check. And this can be demonstrated using mice that are engineered to harbor ablatable Tregs, and this is achieved by inserting a diphtheria toxin receptor under the control of the Foxp3 locus. And in these animals, the Foxp3 expressing Tregs also harbor this diphtheria toxin receptor. And upon administration of diphtheria toxin, this leads to ablation of the cell population. And you can clearly see down here that 2 weeks after continuous DT administration, the mice very quickly develop an autoimmune disease that is very similar to the germline Foxp3 deficient animals seen on the right-hand side. And these mice, within 3 weeks, they succumb to this massive autoimmune activation. So over the years, there has been growing bodies of literature summarizing Treg cell functionality. And now we know that not only are these cells important for controlling autoreactive T and B cells, they're also essential for controlling immune activation to innocuous antigens such as benign environmental and food antigens. They're involved in dampening anti-tumor immunity and controlling antimicrobial immunity. And most recently, they've been implicated in regulating low-grade metabolic inflammation. So dysregulation of any of these arms can lead to many known human diseases of concern, including autoimmunity, allergy and asthma, cancer, infection-induced immunotherapy, such as cytokine release syndromes, and finally, metabolic syndromes, as well as cardiovascular diseases with the underlying low-grade metabolic inflammation. And Treg cells have to accomplish all these tasks while navigating this really dynamically changing landscape of on and off immunological challenges throughout the life of a human being. So obviously, infection and vaccination can happen at any given moment. With age, we know that the chance of developing autoimmunity continues to rise, as is the risk of developing cardiovascular and neurodegenerative diseases. And Tregs have to somehow keep all of these detrimental effects in control while fighting the infection and generating protective immunity that we all need, while suffering from a tremendous drawback, which is that as we age, the thymic Treg output declines very rapidly. And during adulthood, Treg cells are basically lack any meaningful thymic input, and they somehow have to sustain themselves in the absence of thymic output while performing this wide array of tasks and try to optimize the result to keep us healthy. So today I just want to share some of our recent thoughts on how Tregs might be able to navigate these tremendously challenging chores by basically showing 3 vignettes. We want to know when and how much immune suppression is needed, and how do Tregs decide when facing different types of inflammation. And I mentioned that thymic output Treg declines very rapidly during adulthood. And so is there a mechanism for Tregs to maintain themselves and stay competent throughout life? And what is the molecular and cellular basis for that? And finally, I just want to touch briefly on some of our most recent obsession that has something to do with the specificity of Treg-mediated suppression. So basically, the idea is that at any given moment, many of these things could be happening simultaneously. So is there any sort of specificity to Treg-mediated suppression such that it can set different magnitude and threshold towards different ongoing immune responses to make sure the outcome is optimized for each one of them? Okay, so let's dive into the first vignette, which deals with the crosstalk between Treg and inflammation. So based on the mouse models and the human patient data, I think it's pretty clear that Foxp3 expressing Tregs are absolutely essential for preventing the initiation of autoimmune inflammation. However, when I started my postdoc, what was far from clear was whether these Treg cells can function during established inflammation, causing the reversal and resolution. So when Sasha first pitched this idea at me, I was very skeptical, to be honest, because this actually dates back to my PhD work, where I studied the crosstalk between dendritic cells and T cells. So in my PhD lab, there was this very well-known phenomenon where upon activation of toll-like receptors in dendritic cells, they upregulate co-stimulation, and they also upregulate pro-inflammatory cytokines. And one of the mechanisms by which innate immune cells trigger T cell activation was by throwing these pro-inflammatory cytokines at the effector cells, and this somehow made them resistant to Treg cell mediated suppression. And this is a requirement to get productive T cell activation. In MyD88 deficient animals, MyD88 being an essential signaling adapter downstream of the TLRs, you lose the expression of these pro-inflammatory signals. Therefore, the effector T cells cannot overcome Treg mediated suppression, and you don't get productive T cell activation. However, in the MyD88 deficient animals, if you deplete Tregs prior to an immunological challenge, you should be able to restore this immune activation. And this was actually one of the very first experiments I did as a graduate student. And you can see here very nicely in the mediated deficient animals, no or little productive T cell response. And this is restored upon regulatory T cell ablation. And you can see this experiment was done many, many years ago, even before the Foxp3 DTR mouse was widely available to the community. So basically, the bottom line is that pro-inflammatory cytokines can act on the pro-inflammatory effectors, and this somehow makes them resistant to Treg-mediated suppression. And this was something my old lab used to do all the time, and this was something we used to train rotation students, and it worked every single time. So when Sasha pitched this idea at me, I basically said, no, Tregs don't work that way. In the presence of pro-inflammatory mediators, they're going to stop working. And I even found literature that supported my idea because many people working on pathogens had found that pro-inflammatory cytokines induced during parasitic or helminth infection could act on Tregs, causing them to reduce in number or even lose Foxp3 expression and hence their identity and functionality. However, what I was not exposed to when I first started my postdoc was a completely separate field where people tried to use adoptive T cell transfer as a therapy for some autoimmune diseases. And there were actually some at least partially successful examples where adoptively transferred regulatory T cells led to amelioration of various different types of autoimmune diseases, and this included type 1 diabetes, GVHD, as well as autoimmune colitis. And so what seemed to be happening at the time was that there was no consensus in the literature, and depending on the experimental system you use, you seem to get opposing results. So that actually looked very interesting at the time. And this was when I came in and said, oh, maybe it's because of the system people use. And what we need to do is to design a better system to address this question in a clearer way. So what was the problem of the existing systems at the time? So to be able to study how Treg cells function during established inflammation, people had to rely on adoptive Treg transfer. And this system had its merits, but it also had a lot of shortcomings. For example, in any particular disease model, you can only analyze a very narrow spectrum of inflammation. So whatever conclusion you get, it could be specific only to that particular disease model. Second, because it was adoptive transfer, it's intrinsically inefficient, and you have to, once the cells enter the circulation of the recipient mouse, you have no control over where they're going to home to or how long they're going to survive. So during a longitudinal analysis, this becomes even more of a problem because when we do adoptive transfer, we tend to work with small cell number, and over time they get outcompeted by endogenous cells, or they're just not equipped to survive for long periods of time. And so any longitudinal analysis was basically impossible. So to overcome all of these problems, we decided to go ahead and create a brand new experimental system that can bypass the need for adoptive transfer. We wanted to have a system where we can analyze the behavior of Tregs in the context of systemic mixed type inflammatory disease. And we wanted to have a system where we can temporally introduce Treg cells into the disease setting at our time of choice. And we wanted to have the ability to conduct longitudinal observation for as long as we wanted. So bearing these criteria in mind, we realized that the Foxp3 deficient animal itself is actually a great system that shows dysregulation of type 1, type 2, and type 3 inflammation. And we thought that would be a nice starting point. And so with all these criteria in mind, we created this brand new mouse model that expresses a reversible Foxp3 reporter null allele. So basically into the Foxp3 locus, we inserted a stop cassette. Where's my mouse? We inserted a stop cassette shown here that is flanked by 2 LOXP sites immediately preceding the Foxp3 open reading frame. Because of the existence of this LOXP site, Foxp3 transcription is halted within here. So instead of expressing Foxp3, the cell would express this Di1.1 reporter. So we call this mouse Foxp3 lock-stop-LOX, or LSL for short. So if you look at this mouse, instead of a Foxp3 expressing population, all you see is this Di1.1 expressing population. And these are the cells that represent cells that have received all the Treg differentiating signals in the thymus, except that they cannot express the Foxp3 protein. We know that these cells have identical T cell receptor repertoire as authentic Tregs. And in the lab, we call them the Treg wannabe cells. And we knew that these cells had no detectable suppressive activity. And what is nice about this system is that when we introduce an inducible CRE, in this case, CRE-ERT2 controlled by the CD4 promoter, when we administer tamoxifen or 4-hydroxytamoxifen, which is the bioactive form of tamoxifen, we're able to inducibly excise the stop cassette and restore Foxp3 expression in cells that have been programmed to express Foxp3. And at the cellular level, what happens is that the Treg wannabes, upon treatment, are converted into Foxp3 expressing Tregs, shown down here. And I should mention that throughout the study, we used the Foxp3 DTR mice that I mentioned previously, because they express a similar GFP reporter for Foxp3 that allows us to keep track of these cells when we need to. So with this new system in mind, we set out to test whether regulatory T cells are functional during established inflammation. And the way we did it was we took some Foxp3LSL mice, waited for them to become 2 weeks old. And at this point, they were already very, very sick. And we gave them 1 dose of 4-hydroxytamoxifen, and we waited for 4 weeks for the analysis. And as you can immediately see on the right, without Treg restoration, the mice became very sick. They're suffering from splenomegaly and lymphadenopathy. However, with Treg cell restoration, you do not see any of these signs. If we dive into what happened during this 4-week period, what we saw was that very quickly after 4-hydroxytamoxifen administration, within a week, there is a very rapid decrease in terms of activation of both CD4 and CD8 T cells. Also, after 4 weeks, we saw that overproduction of autoantibody, particularly the IgG1 and IgE varieties, were also largely normalized, shown here. I don't have time to show the data here, but we also saw that aberrant activation of the innate immune compartment, such as myeloproliferative disorders and excessive acute phase response, was also normalized within 4 weeks after regulatory T cell restoration. Last but not least, massive immune infiltrate into the tissue, such as the skin, the liver, and the lung. Without Treg restoration, they continue to worsen. However, with Treg restoration, they were also quickly normalized within 4 weeks of time. So all of these strongly suggest that Treg cells are perfectly capable of functioning in established inflammatory environment. However, there's one small dissatisfaction from these experiments, which is that we had to use 2-week-old mice. And it's still possible that this only happens when the mice are still young, the immune system is still being developed, and the regenerative capacity of the non-lymphoid tissues were still high. So is this true during adulthood? Unfortunately, we couldn't just take Foxp3 LSL male mice that are adults above 6 weeks, because they don't live that long. So we have to come up with a workaround. And to do this, we made use of the fact that Foxp3 is an X-linked gene and is subjected to random X chromosome inactivation. So we generated some Foxp3 DTR LSL female heterozygous mice. And what happens in these mice is that due to random X chromosome inactivation, you naturally, these mice become mosaic chimeras. They contain two populations of cells, one expressing the Foxp3 DTR allele, marked green here, one expressing the Foxp3 LSL allele, marked in purple here. And we can take these mice and treat them with diphtheria toxin to deplete the green population. And as shown before, within 7 days, these mice, again, due to the lack of functional Tregs, they become very, very sick, similar to the germline Foxp3 deficient mice. And at this point, we gave these animals tamoxifen to convert the Treg wannabes into Tregs. And basically, when we did this experiment in adult female heterozygous mice, we got the same experiments that we got the same results that we did from the Foxp3 LSL male mice, where the newly rescued Tregs quickly got the autoimmune inflammation under control. And within 5 weeks, these mice completely returned back to normal. So we were very impressed by the powerful ability of these rescued Tregs to curtail an ongoing inflammation, and we wondered what is happening at the molecular level within these rescued Treg cells. So to do that, we took these cells and we conducted some bulk RNA sequencing analyses, comparing them to regulatory T cells from healthy mice. As you can see here, not only are these newly restored Tregs transcriptionally distinct from the Treg wannabe cells, they're also transcriptionally distinct from Treg cells from healthy animals that do not have an ongoing disease. So when we compared genes that are differentially regulated between normal Treg and rescued Treg cells, we saw that the rescued Treg cells upregulated genes that associated with activated Treg signature, and they also upregulate genes that are associated with active STAT5 signaling. When we zoom into this list, we see a lot of these genes encode proteins that are relevant to the suppressive function of regulatory T cells. And just to give you a few examples, these cells become, oh, I'm sorry. I think I may have, oh, I'm sorry. I put the wrong figure here. The bottom line is a lot of the functional genes important for Treg-mediated suppression seem to be upregulated in these rescued Tregs. And based on that, we hypothesize that the rescued Tregs may be functionally superior compared to Tregs from normal animals. So here I'm showing in vitro suppression assays comparing rescued Treg cells with normal Treg cells. And as you can see, rescued Treg cells from diseased male mice are indeed more suppressive than Treg cells from normal mice. And on the right, is the same kind of comparison, but from healthy mosaic heterozygous female mice. And you can clearly see that these populations do not show any difference. So to us, this suggests that these newly rescued Treg cells seem to have the ability to sense the ongoing inflammatory environment. And as a result, they upregulate their suppressive ability to really try to keep this ongoing inflammation under control. And this is even more true when we wait longer, when the disease has subsided. So this is the exact same mouse. We just wait until the disease is completely under control. And now you can see that this difference disappears. Again, suggesting that an ongoing inflammation somehow delivers an activation signal to the Tregs, making them upregulate their suppressive activity. And one of the future directions of the lab is really to understand molecularly what is being sensed and what pathways are being triggered inside the Tregs that makes them execute this enhanced functionality. And to end the first part of my talk, I just want to reiterate that this enhanced functionality of Tregs in an inflammatory environment is not unique to the Foxp3 deficient model that we use, because we see similar phenomena in another autoimmune disease model. So here I'm showing data from an autoimmune colitis model triggered by adding DSS into their drinking water. So usually within 7 days, the animals get really sick, and we did the analysis on day 9, and you can see a lot of the molecules that are associated with Treg cell functionality have been upregulated when at the peak of the disease, when there is ongoing inflammation in the tissue. And we know that at this point, if you withdraw the DSS, these Treg cells can continue to function in this inflammatory environment, and eventually they would keep the disease under control, and the animal would return to health. Okay, so that was the first part of my talk, and for the second part, I want to talk a little bit about our attempts to understand how Treg cells function for a prolonged period of time when thymic input is very limited. And I want to talk about that because we think the Foxp3LSL model that I just talked about could be a great tool to begin to get some ideas about how that could be happening. And so when we did the Foxp3 restoration experiment, the first question I had was, can these rescued Treg cells continue to function? And if so, how much longer can they function? And what do I mean by that? So I told you that we gave the Foxp3LSL mice 1 dose of 4-hydroxytamoxifen when they were 2 weeks old. Normally, if without any rescue, the mice would die. But with this rescue, most of them continue to live, and they can actually live for a very long period of time. And since we know that 4-hydroxytamoxifen has a very short window of action, this 1 dose of treatment would only give you 1 cohort of rescued Treg cells. And so we wanted to know how long can these Treg cells maintain themselves, and can they still remain functional? So we followed the animals for a very, very long time, up to 7 months, at which point we had to sack them because our mouse bill was just skyrocketing. And we couldn't afford to keep them longer, but they could have lived longer had we had more money. So even at this point, 7 months after the single dose of 4-hydroxytamoxifen treatment, the rescued Tregs are perfectly fine, maintain themselves in all of the organs we checked, and the animals were very healthy, no vast difference in terms of immune activation, suggesting not only are they able to maintain themselves, they're also able to maintain their functionality. So we were obviously very impressed by this incredible ability for them to stick around and remain functional for an exceedingly long period of time, because 7 months is a very long time compared to the 2-year lifespan of a mouse. And we wanted to understand their features at single cell resolution, just in case there is some buried heterogeneity that might help us understand their prolonged survival and functionality. So to do this, we decided to introduce a fate mapping allele into these mice. So this is done by knocking in a lock-stop-locks cassette in front of a TD tomato reporter into the ROSA locus. So at the time of 4-HT treatment, in addition to getting Foxp3 re-expression, we're also getting recombination of this locus. So this turns on this TD tomato reporter, and it marks cells that stick around at this moment, and all its progenies in the future with this TD tomato reporter. And you can see here, 1 week after 4-hydroxytamoxifen treatment, the vast majority of the cells are targeted with this TD tomato reporter. And over time, mice that have a normal Treg pool, they continue to turn over and this TD tomato reporter gets diluted, and at the time of the analysis, only 14 to 15% of the cells still remain TD tomato positive, suggesting that these are the cells, these cells are present at the time of Foxp3 treatment 7 months ago, or they are the progenies of those cells, and we term these cells the old Tregs, whereas the TD tomato negative cells, we deem young Tregs because they were likely generated after the TD tomato treatment 7 months ago. And again, and you can see that in the Foxp3 LSL mice, the vast majority of the Tregs still express the TD tomato reporter, again, confirming that they came from 7 months ago, and we call them the long-lived Tregs. And we took all 3 of these populations and sent them for single-cell RNA sequencing. So by this UMAP visualization, I believe what you can immediately appreciate is that the long-lived, the young, and the old Tregs, they occupy different area on the UMAP. And to understand the source of this, the heterogeneity in our data set, we did some diffusion component analyses, and the top diffusion component, component 1 shown here, has a very nice correlation with activated Treg signature, again, suggesting that the major source of heterogeneity in this UMAP comes from different activation status of the cell. So we wanted to see whether these long Tregs are some kind of experimental peculiarity or they resemble something that naturally exists. So we looked at a bunch of parameters. We looked at the value of diffusion component 1. Again, this could be a proxy of their activation status. And these long-lived Tregs, in terms of their activation, look very similar to the old Tregs that are coming from a healthy mouse that is age-matched. In addition to that, we also did an independent trajectory analysis. So this was done using a package called Palantir, which is developed by Donna Perez's lab at MSKCC. So what Palantir does is that it generates a high-resolution pseudotime ordering of all the cells in your data sheet representing their differentiation stage. And by comparing the pseudotime values of the long-lived and old Tregs, you can see that they're highly similar, and they're both above the young Tregs, again, because these cells are more activated. We also looked at other gene sets, such as genes involved in apoptosis, survival, and exhaustion. And in all of these comparisons, our long-lived Tregs looked very similar to their age-matched healthy Tregs from a normal Treg pool with normal Treg turnover. So by all we could tell, it seemed like the long-lived Tregs just behave like Tregs that have hung around for a long time, and this doesn't appear to be an experimental artifact. So this actually sets a very nice stage to study how can these Tregs maintain themselves for so long. And we actually drew some inspirations from a paper from Rafi Ahmed's lab, where they showed that exhausted CD8 T cells are constantly supplied by a stem cell-like population residing in the lymph nodes, and those cells not only are they able to maintain themselves, they can actually differentiate to provide a continuous influx of the exhausted T cell pool. So we're wondering whether something similar exists in our long-lived Treg cell pool. And if so, can we find such a specialized subset that maintains this Treg pool in long term? So to do that, we went back to our single-cell RNA sequencing data set, and this time we did a refined clustering analysis, and one of the clusters, cluster 1, shown here in orange, caught our attention immediately for several reasons. Number one, this cluster is mainly composed of cells coming from the Foxp3 LSL mice. Number two, this population seems to score highly for IL-2 STAT5 signaling, as well as Wnt beta-catenin signaling. So both of these pathways have been implicated in the survival and maintenance of regulatory T cells. And we know that these cells seem to be at a less differentiated stage, as marked by their developmental pseudotime values, suggesting that they have the potential to differentiate into the rest of the Treg pool and sustain them. So by looking at genes that are enriched in this cluster, we came up with a flow cytometry panel that allows us to identify these cells basically in any mouse. So based on the combination of markers we used, interferon gamma receptor 1, EpCAM, and GARP, we came up with a nickname of these cells called gamma regs. So here I'm just showing the gating strategy of this population. And using this flow panel, we did confirm that the gamma regs are more abundant in the long-lived Tregs compared to a normal Treg pool. So that all checks out. So now we want to see whether this population can be found in any normal mouse, and the answer is yes. And not only that, they have a really interesting distribution pattern. So this population is absent in the thymus. So I believe whatever remaining dots we're detecting is just background noise. In the periphery, they are mainly resided in the secondary lymphoid organs, such as the spleen and the lymph nodes, and they only exist in very low numbers in non-lymphoid tissues. And we also noted that this population seems to also be low in the circulation. So does that mean they're less circulatory? And we set out to see if that was indeed the case. So to do that, we set up two assays. One is called intravenous labeling. So for the trainees in the audience who are not familiar with this technology, so what you do is you take the mouse and you inject a fluorescently labeled anti-CD45 antibody intravenously, and you wait for only 3 minutes and you euthanize the animal immediately and collect the tissue. And what happens during this 3 minutes is that the antibody is going to enter circulation. And because we're only treating the animals for 3 minutes, the antibody only has enough time to either stay in circulation or only diffuse for a very short distance into the perivascular space. And this is a way, and this marks all the circulatory cells with this fluorescently labeled antibody, whereas the true tissue resident non-circulatory cells remain IV negative. So by using this technique, we could see that the vast majority of gamma regs are IV negative, suggesting that they're not in the perivascular space and they're sitting deep in the tissue, where they tend to enter circulation much less. And we can even show this functionally by setting up a parabiosis experiment. Again, bear with me if you're already familiar with this system, but for those who haven't been exposed to it, what you do is you take two mice that express different isoforms of CD45 that can be stained with different antibodies, and you surgically join them together. And as the wound heals, the blood supply of these two animals are interconnected. And now whatever circulatory cells can exchange freely between these two animals. And so if a cell type is perfectly circulatory, you would expect 50% of this population to derive from the host, with the other 50% coming from the parabiotic pair. So that is indeed the case for the bulk of the Tregs. However, if we look at the gamma regs, they tend to be much more host-derived, again, suggesting that these cells are non-circulatory and they tend to be more tissue resident. So based on all of the above clues, it seems like the gamma regs represent a secondary lymphoid organ resident resource cells that can maintain the peripheral Treg pool when thymic output is limited, especially during adulthood. And another future direction of the lab is to understand functionally how this population maintains the peripheral T cell pool and how does it sense the signal that thymic output is limiting and it's time for them to do their job. Okay, so for the last 5 minutes or so, I just want to share very briefly about a new obsession of the lab. This is a very ambitious endeavor. I'm not even sure if it's going to work, but I think it would be a very interesting question to tackle if we have the right kind of tools. And this has something to do with the antigen specificity for Treg cell mediated suppression. So I think we can all agree that T cell receptor is absolutely essential for the differentiation of Treg cells. And most recently, people have also tried inducible deletion of the T cell receptor in developed regulatory T cells and showed that the T cell receptor is constantly required for the suppressive activity of Treg cells. And what I was interested in is, does the specificity of these TCRs matter? Or is it that any sort of tonic TCR signaling would do the trick? So to begin to tackle this question, we actually started with some relatively simple experiment using a mouse model that we created for a completely unrelated reason. So this mouse, we call it the ROSA Foxp3 mouse, where into the ROSA locus, there is again, a lock-stop-locks cassette. And upon induced excision of this lock-stop-locks cassette, you can basically force Foxp3 to express in any CRE expressing cells. So initially, the motivation of creating this mouse was to study a newly discovered suppressive T cell population in the germinal center that I don't have time to go into. But it turns out that this tool is also valuable for tackling the question of Treg TCR specificity and how it's linked to their function. And what do I mean by that? So what we can do is we can breed this mouse into a mouse where the Treg wannabe cells harbor a reporter that we can sort with. And so in this scenario, we have the Treg wannabes, and among the conventional T cells, we can then separate them further into the naive and memory populations. And we can take these three populations separately, turn on Foxp3 ectopically from the ROSA locus, and we can then put these cells into mice for an in vivo suppression assay. And what you can see here, I think this is a very interesting piece of information, is that as you would expect, without Treg, you get massive accumulation of proliferating T cells, which is suppressed by authentic Tregs. And Treg wannabe cells, when supplemented with Foxp3 coming from the ROSA locus, can also do the same thing. What is really interesting is that if you put Foxp3 back into naive or memory CD4 T cells, they have no ability to do so. And this was somewhat surprising to me because if you look at molecularly what's happening between these Foxp3 supplemented T cells, they're not that different. So obviously, without Foxp3, they start out being very different. Also, I'm sorry, here we're looking at an ATAC-seq experiment where we're looking at chromatin accessibility in all these comparisons. So in the absence of Foxp3, these three populations are quite different. With Foxp3 expression, they're brought a lot closer and they're all being pushed to something like an authentic Treg cell. And this remaining difference, we think, is mainly because a slightly lower expression level of Foxp3 expressing from the ROSA locus versus Foxp3 expressing from the endogenous locus. And it almost feels like as long as Foxp3 is expressed, it doesn't matter what cell type it's expressed in. And we actually know that if we take these cells and we perform an in vitro suppression assay, they're equally suppressive. But somehow only the Treg wannabes are suppressive in an in vivo setting. So this is an intriguing piece of data, and there could be millions of other explanations, but based on what we know about these cells and other data that I don't have time to go into today, what we believe is happening is that the Treg wannabes work better in vivo because they bear a unique TCR repertoire that guides them to the right place and do the right thing. However, when you do an in vitro suppression assay, this uniqueness is bypassed because in an in vitro suppression assay, you don't engage the actual TCR. You use a pan TCR activating antibody that is anti-CD3. All right, so to summarize what I've shown today, I hope I've convinced you that Treg cells are functional in systemic mixed type inflammation that includes type 1, type 2, and type 17. And not only are they functional, they seem to have the ability to sense an ongoing inflammation, expand themselves, and upregulate their suppressive activity. I showed you that a single cohort of Treg cells can stably persist and provide long-term protection against autoimmunity. And based on our single cell analysis and flow analysis, we think that this is potentially mediated by specialized source cells that reside in the secondary lymphoid organs in the periphery. And lastly, we are beginning to gather some clues about the role TCR specificity plays in Treg cell functionality. And we think that the specificity truly matters, especially in an in vivo setting, not because molecularly what happens in terms of gene transcription, but because they have to be directed to the right APC and the right microenvironment to do their job. Okay, so with that, I'd like to thank my past mentors, Sasha and Shaker from MSKCC and Cincinnati Children's, respectively. Past and present collaborators, the Victoria Lab, who I really enjoyed working with, and our current collaborators who are helping us with bioinformatics analysis, YREG's lab and Matt's lab from Yale. And finally, a huge shout out to the Yale West Campus, where people from, by the way, this is where my lab is located. So there's something truly unique about Yale's West Campus. People from different fields really try everything to come together with incredible collegiality and do the best science possible. And with that, I'd like to take any questions. Thank you.